All right, I see people are coming in from the waiting room. Welcome everyone to the Research Brown Bag. I'm Jenny Hearn, Associate Dean for Research, and I'm really pleased to introduce our speaker today, John Marshall. He's an Associate Professor in Biostatistics, and all of his work is around strategies for mosquito control and in an effort to reduce the burden of malaria. So John, I look forward to hearing about your work, and thanks for joining us.
Okay, thanks Jen for the introduction, and thanks everyone for being here. I just wanted to give a bit of an overview today. This topic of mosquito genetic control is not something that most people in the school work on. So I wanted to give a broad overview of the field as a whole and the current status of the various projects to control mosquito-borne diseases using these tools, and throughout that description, then there will be pieces of our modeling work. Our group works on mathematical modeling of mosquito-borne diseases and genetic control strategies. So throughout this overview of the field, I'll have a description of some of the projects that we're working on as well. So I wanted to begin by just giving a little background on the global burden of malaria, dengue, and other mosquito-borne diseases, and the need for novel tools to control them. Then an overview of the current status of some of the strategies to control mosquito populations in a self-limiting way, based on the sterile insect technique. Also some strategies which incorporate Wolbachia transfection, which are also being implemented here in California. And then some of the strategies which are a little further off to introduce mosquitoes capable of spreading disease-refractory traits or genes into mosquito populations and controlling disease on a wider scale in that way. Also want to talk about some future project needs in terms of modeling, community engagement, and regulation. And this work, when I refer to our work, is collective work of our research group. So I just wanted to acknowledge them, and it's been a great pleasure to work with them over the last 5 or 6 years in the school. So to begin with, the global burden of malaria, and this, as you can see on the left, this is data taken from IHME. The burden of malaria in terms of deaths was rising up until shortly after the year 2000. And then there was a large investment in wide-scale interventions to reduce that disease burden. And since that time, there has been a reduction through, largely through the distribution of insecticide-treated bed nets, also spraying of walls with insecticides, and distribution of antimalarial drugs. And the burden of malaria deaths, which is over 90% in sub-Saharan Africa, has been declining. It has actually been trailing off recently, and the map on the right shows model predictions of locations in Africa where we would expect these currently available tools, the insecticide-treated bed nets, the spraying of walls with insecticides, and antimalarial drugs, to be sufficient to eliminate malaria. And the region in gray, which is of relevance to this talk, is where it's not expected that these currently available tools will be capable of eliminating malaria. So we're in need of additional tools to complement those that are already available. And then in terms of dengue and arboviruses, chikungunya, Zika, particularly dengue, in the chart on the left, it has been growing in case numbers over the last couple of decades. There have been some larger and larger outbreaks that have been occurring in several regions throughout the world. There have also been additional outbreaks from chikungunya and Zika And the mosquito vectors that spread these diseases to humans have been expanding in range. This is Aedes aegypti and Aedes albopictus. And just in the last 5 or so years, they've been expanding in range to California. So the mosquito species that could transmit these diseases are now present here as well, which wasn't something that was the case, more than 5 years ago. This could be due to climate change and also adaptation of the mosquito species to new environments. So moving on to some of these suppression strategies for mosquitoes that revolve around genetic control. The original genetic control strategy was the sterile insect technique, which revolves around mass rearing mosquitoes, then irradiating them with sublethal doses of radiation, and separating the males from the females. It sterilizes the males due to the irradiation. Those males are released, they mate with the wild females, and this wastes the reproductive potential of those females in the wild, which results in the population size declining over time. And the most famous application of this technique was to the New World screwworm, which through the 1950s to early 2000s, progressive sterile insect campaign released these flies and eliminated them from North America, with the boundary progressing southwards towards South America. And to this day, there are still large-scale releases of New World screwworms that are irradiated and sterilized through the irradiation at the border between South America and Central America. So this is a precedent that similar things could be done with mosquitoes were their interest in investment. And the International Atomic Energy Agency has invested and collaborated with a lot of groups. On this slide, there's a depiction of the timeline for SIT program for Aedes albopictus, the vector of dengue and other arboviruses in Reunion Island, where small-scale pilot field trials are currently underway and moving towards large-scale field trials. But these trials have been happening since the 50s for mosquitoes. They're now moving forwards with new technology. Part of the projects are to tailor the irradiation so that the males that are sterilized are sterile, but they're still viable and they still have competitiveness to mate in the field. An advancement from SIT has been genetic versions of this, which revolve around transgenes rather than irradiation. One of the problems with irradiation is that it can reduce the fitness of the organisms that are subject to this radiation. And so one of the ideas of Luke Alfie, who started a company called Oxitec Limited in the UK, was to have a gene that conferred lethality when present in 2 copies, or actually no, when present in 1 or 2 copies. And this was under the control of a promoter that could be turned off in the presence of tetracycline, an antibiotic. And so in this way, you could rear mosquitoes, separate the males, and in the presence of tetracycline, they would grow and be viable. Then you release them into the wild, they mate with the wild females, but without the presence of tetracycline in the wild, then you end up with a population suppression occurring. And there have been several field trials of this technology. So this is one shown that took place in Bahia in Brazil in the first half of the last decade. And we see here that it was an interesting field trial where they had a range finder period to begin with, where they released a certain number of these genetically sterile males. And by seeing how much the population declined during that initial phase, they figured out what the mating competitiveness of these released males was relative to the wild males. They actually found that the mating competitiveness was about 5% that of the wild males, which is a significant reduction in mating competitiveness, which needs to be improved upon in future versions of this technology. But as a result, for this field trial, they created a smaller target area. Initially, they were trying to control areas A and B. They moved just to area A, and they increased the density of released sterile males by 20. So 20 times more genetically sterile males were released. And that resulted in the suppression that you can see in panel D here. But there are still issues related to leakage of the toxin, which reduces the competitiveness of these mosquitoes, which future generations of this technology will improve upon. There are also technologies based on Wolbachia infection. Wolbachia is an intracellular parasite that's present in about 50% of insect species. It wasn't present in Aedes aegypti, which is the major mosquito vector of dengue, Zika, chikungunya around the world. And so when introduced, it alters the reproductive biology of the mosquito. So if a wild male mates with a wild female, the offspring are viable, of course. If a male with Wolbachia mates with a female with Wolbachia, then all offspring have Wolbachia. If a male without Wolbachia mates with a female with Wolbachia, then all of the offspring are viable. The Wolbachia is passed on to the offspring. But if a male with Wolbachia mates with a female without Wolbachia, then something referred to as cytoplasmic incompatibility occurs and the offspring are inviable. So this cross here of males with Wolbachia and females without was used as a basis for what's called the incompatible insect technique, where if you release males with Wolbachia into the population, but no females, then all of the matings between those mosquitoes will be inviable and you can get population suppression akin to the sterile insect technique. I'll talk about the population replacement strategy later. But this technology has been implemented. There was a company called Mosquito-Mate that started out of the University of Kentucky, I believe, and they formed a collaboration with Verily Life Sciences based in South San Francisco, the Debug Project, where they mass produce in a very automated way male mosquitoes having the Wolbachia infection. And they separate by sex. It's very important for this technology that you only release males. So they can do this by separating at the pupal stage. Males tend to be smaller in terms of pupa. They have a number of other stages, some of which involve machine learning by photographing the mosquitoes as they walk through a chamber, and you can see the male and female mosquitoes. The females have a wider abdomen because they have to produce eggs. The males have sort of a bushy antenna, which have been compared to a mustache. And through multiple stages of sorting, they can reduce the error rate to less than one in a billion, or several million at least. So that enabled them to produce large numbers of Wolbachia infected male mosquitoes. And they carried out a trial in the last few years in Fresno, Clovis, California. You can see the control areas and the treatment areas, C1, C2, C3, T1, T2, T3. And the Aedes aegypti population in Fresno is seasonal due to the weather in the region. But you can see that the control areas had a large population size increase, whereas the treatment areas remained at low population. There was a population reduction of around 95% through the widespread release of the Wolbachia infected males. And this was also carried out an analogous control program in Guangzhou in China. There was, in Guangzhou, they were implementing this in Aedes albopictus, which is another vector of dengue, Zika, other arboviruses. These were already infected with some strains of Wolbachia, so they added an additional strain of Wolbachia for the released males, which ends up having the same cytoplasmic incompatibility result. And releasing males with these strains of Wolbachia, when they mate with the wild female mosquitoes, produce no viable offspring. To prevent the release of viable female mosquitoes in Guangzhou, they combined the Wolbachia transfection with the sterile insect technique. So they actually irradiated the mosquitoes as well. And this prevented the Wolbachia being passed on through the female mosquitoes to the wider population. If that happens, it can prevent the ability to suppress the population. So they combined the sterile insect technique with the incompatible insect technique, and you can see here with the suppression efficiency nearing 100% in both of their treatment sites relative to their control sites. So that's been a very effective strategy as well. A version of this technology that we are working on has been made possible thanks to the CRISPR revolution and Jennifer Doudna and colleagues in the building usually next to us, where our colleagues at UCSD, Omar Iqbari's lab, have combined mosquitoes having the Cas9 protein and mosquitoes having guide RNAs which target alleles required for female viability and male fertility. So when you combine the guide RNAs and the Cas9 in the same, when you mate those lines together, produces organisms which, if female, are unable to fly, and if male, are sterile. And the benefit of this technology is that you can combine these lines and produce eggs, and Aedes aegypti eggs can be stored for very long periods of time. Not sure the exact time, but it's on the order of years. And then once they're put into water, then they emerge. So you can store these eggs and then release them, deliver them into the environment, and they would be sterile males, which when they mate with wild females would produce unviable offspring. Our modeling work of this technology suggests that it could be more efficient than some of the competing technologies, because at the larval stage, there's an additional density-dependent mortality. So if you have more larvae in the breeding sites, then they compete with each other, and it reduces the population size of the wild larvae as well. And this happens early on after a release, which can cause a quicker decline in the population. Also compared to the RIDL construct, the genetically sterile males mentioned earlier, those have a mating competitiveness reduction, which we think could be avoided with the PGSIT approach. So we've been modeling these and other mosquito control interventions using Mosquito GeneDrive Explorer modeling framework. It has a number of modules. There's an inheritance module which models the passing of genes from mother and father to offspring, and how that is varied depending on whether you have a sterile technology or an inheritance biasing technology. Then we follow the mosquito life cycle from egg to larva to pupa to adult. We also describe the distribution of mosquitoes in space and their movement through space, which is important for understanding how genes disperse through environments. And then we have an epidemiology module, which describes the transmission of disease in both humans and mosquitoes and the transfer of pathogens between the two. And here we have a simulation. This is a visualization thanks to Hector Sanchez and Jared Bennett, especially Hector Sanchez, who's our data visualization whiz. We're currently discussing releases of PGSIT technology in Onatahi in Te Aroa, French Polynesia. It's actually a site where the incompatible insect technique has been applied in the past. And we've simulated here releases at multiple sites throughout the island. On the graph in the middle, you can see different numbers of eggs being released per weekly release, and then the number of consecutive weekly releases that are carried out. And we simulate the release sizes that can lead and durations that can lead to population elimination in these settings. These are preliminary simulations, but they will be improved upon as we learn more about the ecology of Aedes aegypti in these settings. But you can see the sort of range of release parameters which lead to elimination or expected elimination shown here. So we expect that on islands like this, we might actually be able to eliminate Aedes aegypti using this technology. Okay, I wanted to transition here to talking about technologies which are self-propagating or capable of spreading into a population from an initial release. And Wolbachia actually provides an opportunity to do this as well as suppressing populations. So the Wolbachia actually can be used to either suppress a population through releasing males which have Wolbachia, and due to this sterility of the resulting cross with wild females. But if you release Wolbachia infected females, then all of their offspring pass on this Wolbachia infection to the offspring. And this actually biases inheritance in favor of Wolbachia, because all offspring involving one Wolbachia parent are either inviable in one case, or it's passed on in every case in the other, which biases inheritance in favor of Wolbachia over the wild type non-Wolbachia state. So if we include females with Wolbachia in our releases, then we can actually have the bacterium spread into the population rather than suppress it. And something interesting about Wolbachia is that it's been shown to reduce the transmission of dengue and other pathogens to humans. It's thought that perhaps it upregulates the immune response of the mosquito because it's constantly fighting off an intracellular bacterium. And as a result, it also is ready to fight off dengue and other arboviruses. There are other theories as well. But in doing so, by releasing females with this Wolbachia bacterium into the population, then we can spread an arbovirus refractory trait into the population. So this actually has been carried out about 10 years ago now. And this is a depiction of one of the field sites that was used, Yorkeys Knob in Queensland, Australia, where weekly releases were carried out over a number of months. And you can see here the weekly release sizes in the gray bars, and then in green, the proportion of mosquitoes that had the Wolbachia infection increasing over that period as well. So this was effective at reducing dengue transmission. Dengue wasn't endemic in this region of Queensland, but there were occasional outbreaks after introduction into the population, and it's prevented dengue outbreaks from occurring since the intervention happened. Something that we've been interested in from this technology is just, and these trials, were the heterogeneity of spread into the population. You can see here different frequencies of Wolbachia at different time points, and the spread isn't homogeneous. So when we want to model the spread of other genetic control tools into mosquito populations, we really have to understand this technology. It can help us to inform the heterogeneity in our models and the monitoring strategies that can ensure that we reach all of the households in the community to prevent their exposure to disease. Nick Jewell and Suzanne Dufault in our school were involved in a epidemiological outcome trial of this technology in Yogyakarta. And this was one of the first trials of a genetic control tool in mosquitoes where the outcome was actually epidemiological. So it's a really exciting result. You can see that Wolbachia infected mosquitoes, Aedes aegypti, were released, and the Wolbachia bacterium spread into the population. And then you can also see that there was a decline in the dengue incidence following the intervention. And they used a test negative design to show that there was a statistically significant reduction in dengue incidence as a result of that. So Wolbachia is an interesting case study for GeneDrive technology, because in the case of Wolbachia, you're actually driving intracellular bacterium into a mosquito population, and it's having this impact on reducing disease incidence at the same time. And GeneDrive technology, the goal is to do essentially the same thing, but using transgenes rather than this intracellular bacterium. One of the issues with the bacterium is that it's an entire organism. So you're introducing an entire organism into another organism rather than just introducing an isolated gene, which you have perhaps more control over. Although it does involve using transgenes, which are controversial and have issues associated with them as well. So this technology has really been advanced hugely in recent years through the advent of CRISPR gene editing technology. Essentially, you have the GeneDrive system on one chromosome, and it can cut a site on, if you have a heterozygote where one chromosome has it and the other doesn't, it can cut the opposite chromosome at the site where it is, and then serve as a template for repair, copying itself to the opposite chromosome as well, and essentially converting a heterozygote into a homozygote for this gene. If you combine this gene with a disease-refractory trait, then you could spread that disease-refractory trait into the population, which is sort of shown here, where all the offspring of a cross between an organism having the GeneDrive system and not will result in organisms having it. And there's been, in some of the work that we're doing with the UC Irvine Malaria Initiative, there's been a lot of progress in this technology. They have engineered GeneDrive systems with red eye markers so that you can monitor their spread into mosquito populations. And here you can see 1-to-1 male releases and 1-to-9 male releases, and the GeneDrive system within, say, 7 to 8 generations spreads to fixation in these populations, which is encouraging that, perhaps even within, if you had a small trial site, then within the timescale of a year, you could spread this trait into the population or less. In addition to studying the laboratory performance of these technologies, then it's crucially important to understand the ecology of the mosquitoes that you're working with. So we've spent time on some of the island field sites where we're working to characterize the distribution of mosquitoes and later their movement patterns. And correlating this with GIS data on land cover type, on distribution of roads, of rivers, and population density to help understand the distribution of mosquito population throughout the islands. Mark release recapture experiments and genetic strategies are being used to understand how mosquitoes move throughout space on these islands. So if we want this allele to spread through a population, then it's crucial to understand how much mosquitoes move, and sometimes understanding how existing genes in the population have moved through those populations is the best precedent for understanding how an introduced gene might spread through the population. As we have these models calibrated to these release sites, then we can simulate potential intervention scenarios, field trial scenarios, also biosafety features of if we want to contain a release to a certain area. So here we have simulated, thanks to Jared Thomas and Hector and others, a release in the south of the island of Sao Tome, releases at several sites throughout the island, and a release in the capital. And we can see how we might expect the trait, the anti-malaria trait in red, the wild trait in blue. We can see how we might expect the anti-malarial trait to spread throughout the island following releases of different types. There's a number of suggestions of how these interventions could be carried out. It's possible that you might do a seeding release in, say, the south and get some understanding of the spread, and then take some of the larvae from those sites and introduce them into other locations throughout the island. Another strategy which would spread genes into mosquito populations aims not to introduce disease-refractory traits, but rather to introduce traits that would induce a fitness load or a sex bias in the mosquito population. So one approach here would be to interfere with a gene that's required in at least one copy for female mosquito fertility or female mosquito viability. So if you introduce those traits into the population, you can eventually have them spread into the population and induce a population crash by causing the females having the gene not to have any offspring. One of the issues with this approach is that resistance alleles, so if you have the GeneDrive construct cut the opposite chromosome, serves as a template for a pair, but then you have a error-prone copying event occur, you can have an allele form which can retain the fertility trait but block the GeneDrive from registering that and recognizing that site in the future. So if these resistance alleles emerge, then they can be selected in the population because they're more fit than the original wild type gene, and they can disrupt the success of this approach. But there has been some progress. These are our colleagues at Target Malaria, and they have actually discovered a gene called double sex, which is required in sex determination in a number of insects, including fruit flies, but also in mosquitoes. And this allele is very conserved, even more than would be expected due to the redundancy in the genetic code, because it seems that there may be just the genetic structure of that allele is required to be conserved for it to be recognized. And resistance alleles have not been detected, and the GeneDrive construct has been working. This GeneDrive construct targets a gene required for female sex determination. So it results in females being intersex male or infertile males, essentially, shown here. And so it drives the population towards one in which you have males and infertile females, and you get a population crash, which has been seen in the laboratory and occurs in, on the scale of 8 to 12 generations, which is encouraging. But this experiment was conducted for a population size of around 300 mosquitoes, whereas in the wild, of course, you have a lot more than 300 mosquitoes. So there's still potential that resistance alleles could crop up in reality. And these releases have been simulated on the scale of potential interventions. So one of the field sites which the Target Malaria project has collaborations is Burkina Faso. They've divided Burkina Faso up into grid cells, incorporating some geographical information, then simulated releases of this double-sex gene drive system. And they've labeled a few locations here on the map where they've shown the outcome of these releases. An interesting feature of this approach is that it does result in population suppression. These areas in green are where the mosquito population has been completely eliminated. But then there's a possibility of recolonization of mosquitoes, which are viable and from neighboring sites. And so they can recolonize, but then to be suppressed again. So it's still up for debate whether this is a preferable approach to the population replacement one where you introduce a disease-refractory trait, which is the approach that we're working on. The disease-refractory trait approach has the issue that the malaria parasite could evolve resistance to the introduced trait, whereas in this case, there's this extinction recolonization dynamic and also the resistance to the female infertility trait. A couple of directions that we're moving into regarding modeling are target product profiles. For one, we have a project that's just about to begin with the Bill and Melinda Gates Foundation, and they're interested in understanding the parameters of genetic control tools, which would lead to them having the intended impact. Some of these are ones that you could measure empirically in the lab, and some of them are ones that you would need to extrapolate from the lab parameters. And one of those is the impact of the technology on malaria transmission. So we have included here some simulations where the genetic trait spreads into the population. We've incorporated some weather and climate information, and also the impact this would have on human disease prevalence and incidence. There is some debate in the community over what sort of reduction in disease incidence would be required. And for the rollout of insecticide-treated bed nets, they set the goal relatively low at 20 to 30% reduction in clinical malaria incidence. And this was because they wanted to show in a trial that it would have the absolute minimum impact. And they didn't want to set the bar too high, because then if the trial failed, then we wouldn't have seen the rollout of insecticide-treated bed nets that we have seen over the last decade, and which has caused a massive reduction in malaria incidence. But with this technology, because the stakes are higher, because there's more controversy, and it's expected that the required reduction might be higher, say around 50% reduction. There might still be transmission because there are other mosquito species and complications that could lead to ongoing transmission. Another area which the Bill and Melinda Gates Foundation want us to look into is monitoring and surveillance. And so the heterogeneity point and spread, which I mentioned earlier, is relevant, but also surveillance. One of the perspectives of the Gates Foundation is that this project is going to be something which, if it occurs, would occur over the timescale of decades, and they want to continue their investment over that time period. And they expect that monitoring surveillance to be the cost driver in after release. Once you've already developed the technology and the logistics of carrying out the releases, then monitoring on such a large scale, national scale, regional scale, to make sure that things are proceeding as intended, and that negative impacts are not being seen, or the technology isn't failing, then that requires a large effort. And they've tasked us with understanding the best distribution of traps and analyzing that data to best inform that. Actually, I want to leave a little bit of time for questions, so I'll just run through these last slides relatively quickly, but risk assessment is also something which is proceeding very quickly at the moment. And there was actually a paper published just a couple of days ago here with some risks of this technology which should be explored, and some of which could involve a modeling-based assessment. Particularly if you release mosquitoes with the goal of reducing malaria transmission, there's a concern that there might be other diseases such as Onyonyong virus and lymphatic filariasis, which could be transmitted. Modeling should help to assess whether it's a concern that those could increase as a result as well. There are many other risks, and those would be populated through a general risk assessment involving a whole range of stakeholders. Community engagement, of course, is absolutely essential and is of growing importance for this project. The Target Malaria project, thanks to funding from the Gates Foundation, there's been a huge investment outweighing even the technical aspects of the project and community engagement. And there has also been a large effort towards capacity building and engaging with scientists in disease endemic countries. So there are some recent papers that have come out on the linguistic process of how Target Malaria approaches this, and work in this area is ongoing. In my own experience with spending some time in Mali and engaging with community in these projects, there's a large interest in them because malaria is a huge burden, and almost everyone knows someone or a family member or friend who has died of malaria. But at the same time, they want to see evidence that this technology is going to have the impact that is that's being stated, and so that requires a lot of community engagement and collaboration. There are interesting precedents from other genetic control interventions. The release of genetically sterile males in Brazil that I mentioned earlier, which required the 20-fold additional release. They had a very comprehensive community engagement program involving community outreach, public talks and lectures, collaborations with the local organizations were driving the intervention. But an issue arose after the trial that there was the Zika pandemic, and there were rumors that the Zika pandemic had been caused perhaps by the release of these genetically sterile male mosquitoes. And this did put into question whether such releases would be supported in the future. So it stresses the importance of a comprehensive community engagement process. And actually, one last thing that I would mention is there's of course also regulatory concerns and the potential of technologies to spread across national borders, which would have to be assessed. And so those regulatory considerations are also up for of great importance for a discussion as technology advances from the lab to the field potentially. So just to recap quickly, malaria, dengue, and other mosquito-borne diseases continue to pose a major global health burden. There's a need for new tools and transformative tools to control their spread. There have been a number of trials and interventions with these self-limiting genetic control tools in recent years, and a number of options have been emerging, including here in California. There's also been recent technological progress thanks to the advent of CRISPR gene editing on self-propagating mosquito genetic control tools, and discussions are moving towards field trials. There are a lot of issues that need to be addressed, and further research is required in terms of modeling related questions, community engagement, and regulation. I'd just like to thank our funders and once again our lab members, and maybe we have just a few minutes for questions.
Great. Thank you so much, John. People want to pipe in with questions or type questions in the chat. Here is one from Sally. Thank you for this interesting talk. Of course, mosquitoes are a pest for us even when they don't carry disease, but is there any conservation concern about dramatically reducing the population of specific insects? It seems like it could affect the population of other animals, including birds, lizards, and frogs that eat those insects, potentially affecting the whole ecosystem.
Yeah, thanks, Sally. So there is concern about all of these things, and yeah, it depends on the location where you are. One of the benefits of this technology is that it's very specific to an individual species, so we could potentially, reduce or replace the major mosquito malaria vector without reducing populations of other insects. Of course, insecticide-based tools like DDT and others that are used today, pyrethroids, reduce a whole host of mosquitoes. So in some ways, this might actually be a more environmentally friendly technology. But that said, when we're considering risks, we want to consider all possible outcomes that could occur and then to study them very systematically. So the risk assessment paper that just came out a couple of days ago does detail these. There's a list of, I think, 48 risks, and each one, it has a page of the risk hypothesis and points where we would want to investigate some of these questions. So if risks appear to be very realizable, then that would warrant scientific study, ecological study of the relevant populations.
Other questions?
Well, this is so basic that I hesitate, but if nobody else is asking, I have trouble understanding with gene drives how something that doesn't confer a selective advantage ends up dominating the population, and something that is causing sterility or otherwise, you would think would be not conferring a competitive advantage on the individual with those genes, therefore shouldn't be expanding. I continue to not understand that.
Yeah, so this is, yeah, it's a question of whether the inheritance bias outweighs the fitness cost. And in this case, the fitness cost is only manifest in homozygotes, so organisms having 2 copies of the gene, and females. But in males, there's no fitness cost, heterozygotes or homozygotes, and in females that are heterozygotes just having 1 copy, there's no fitness cost. So the gene can sort of sneak into the population through these other genotypes and only manifest in homozygotes. And in that case, the inheritance bias can outweigh the fitness cost and lead to suppression or elimination.
So my dumb correlate with that is sickle cell, but there the heterozygote has a fitness advantage in certain contexts, which for me explains why it became so prevalent. When it doesn't have a fitness advantage, why would it increase?
So it doesn't have a fitness advantages, but it has an inheritance advantage in the sense that you're converting a heterozygote into a homozygote in both males or females.
All right, I know we're out of time. I need a little more tutoring.
Well, thank you so much, John, for sharing your work. It's really fascinating and yeah, super interesting. So thank you so much for your time, and thanks everyone for joining.
Okay, thanks. Yeah,
Have
thanks,
a good
everyone.
day. Bye. Take care, everyone.